to?
Kristen, are you the one that has the slides?
Kristen.
I'll be sharing my
Hi,
screen.
Dr. Brooks. Yeah, it's going to be Amanda with the slides.
Okay.
Amanda.
Make Amanda the co-host here. Okay, Amanda, you're all set. Or you should be all set.
All right, so today Amanda, Alex, and I are going to introduce Dr. George Brooks. A little bit about him is that he has a wife and 2 sons along with 2 grandbabies. He is a cyclist and a skier. He used to run track and field as well as cross country. He's currently thinking about joining a softball league, but he's not really sure if he has the time for it, and you'll see why coming up in a few slides. So some fun facts about him. His wife is actually a sports medicine doctor and is one of the 3 doctors who coined the term the female athlete triad. He's currently writing a chapter for a book called The Frontiers of Physiology. When he was finishing his postdoc, he applied for the only job available in the country at the time, and he got the job, and he's actually been working there ever since. He's the former president and vice president of the Southwest chapter of ACSM. And then he has a ton of areas of interest, and they include these, but they're not limited to metabolism around glycolysis, lactate metabolism in cancer and traumatic brain injury, obesity and diabetes, lactate shuttle, aging biology, and menopause.
Alrighty, and now I'm going to get into Dr. Brooks's educational background. So for his bachelor's degree, he went to Queens College in New York, which he referred to as the poor man's Harvard. He got his master's and PhD at the University of Michigan in exercise physiology. And he got his postdoc from the University of Wisconsin in muscle biology. So Dr. Brooks has many publications and citations. He has over 300 published works and has been cited nearly 40,000 times. He also has an H index of 102, and I threw a picture of his exercise physiology book down there, which I thought was cool. All right, and Dr. Brooks has been given several awards. He was given the Honors Award from the College of Sports Medicine and was an honorary professor from a college in France, as well as an honors award from the ACSM. And Dr. Brooks is a professor at the University of California Berkeley, and he's been teaching there since 1971, which is very impressive. From last semester to current, he has been involved in several classes. As we see, he's been in integrative biology, he does graduate research, and occasionally does seminar and human physiology courses.
Dr. Brooks has received funding from the National Institute of Health for over 30 years. And the research that the funding that he's received amounts to about $4 million. He's currently researching metabolism with his lab, and he's previously received grants from his research in MCT gene expression and exercise substrate utilization. These are just some of the works, a little bit more recent works that he's received funding for. When Dr. Brooks works with graduate students, he likes to take a hands-off approach so that they can become independent researchers, but he does tend to keep contact with them after they graduate. Dr. Brooks has mentored both master's students and PhD students. Most of his students come from the CSU system, just like us. And his very first PhD student became a chancellor of the CSU system. So when we asked Dr. Brooks how he became successful, he told us that he was initially motivated for graduate school by an exercise physiology instructor. Interestingly, he is the first person in his family to graduate from high school, let alone college. And throughout his education, he never felt like giving up. He says that motivational drive is essential to become successful. When we asked Dr. Brooks what advice he would give students, he really emphasizes having a genuine interest and passion for the field that we get into so that our hobbies can become our careers. Dr. Brooks suggests that we look into journals that we would like to be published in, and then find out where those people are working, and then to find our mentors there. And he advises students to work hard, but not too hard. Presenting Dr. Brooks.
Thank you. That was quite flattering. I'm happy to talk to young students like you because I remember still when I was one. So how do we proceed from here?
Well, I think a good place to maybe just start a little bit is I know you were a talented middle distance runner and cross country runner. And so did that kind of fuel your interest in human physiology a bit? Or how did you end up going down the biology route?
Yeah, well, I think when I talked to Kristen, I did let her know that I wasn't as successful as I wanted to be. So, you know, in sports, you stand on the boxes, right? And I never got to the top box. So I wanted to understand why. And that's what created my interest in physiology. And once the person who advised me as an undergraduate was Bill McCardle of the textbook McCardle Catch and Catch, and he set me up to go to the University of Michigan and helped me get a fellowship. And after that experience, which was transforming, my life was just that way. Working in a lab, doing experiments, and even today, you know, we're doing. Right now, outside of my office, I have a technician, a postdoc, and a graduate student all working on a mitochondrial prep from a very old animal. So we're studying mitochondrial energetics and aging. So it's really cool stuff. Work with young people.
Did you ever, in your career, did you ever measure your VO2 max? And if you did, what was it? You seem like a pretty athletic person, so.
Well, it was 4.5.
Ooh, nice. Leaders, nice. Okay.
Now it's 2.2.
Oh, okay. That's still pretty good.
Yeah, so that's and that was, you know, when we were in graduate school, if you had an experiment, I was your subject, and then you were a subject in my experiment, and we really learned from the ground up.
Yeah, I remember. So for you, the students, my master's mentor up at Chico State, Jack Azevedo, did his PhD with Dr. Brooks at Cal, and Jack did his bachelor's, and I think he did his master's at San Francisco State. And one of the projects he was drafted into was running it might have been one of your projects, George, the marathon on the treadmill. Was that one of yours?
Yeah.
Running a marathon on the treadmill in the lab and giving blood samples and doing all kinds.
You know, that was really one of the beginnings of the crossover concept. So people had the idea that if you're going to be a good distance runner, you're going to use fat. And I said to myself, nah, that's not right. So people ran on a treadmill. We got a brand new treadmill. and was Mark O'Brien. He was a master's student from Australia, and Mark was if we have any 800 meter runners in the group, he was 144, which is still world-class time. And so he hadn't trained in 2 years. So we got this new treadmill. He said, I think I can run a marathon. So he just jumped on it, ran a marathon. Just cold. He hadn't run for 2 years. And then so we measured his gas exchange R, and you can see that, it was 0.95 until he got tired at the end and tailed off. So that was really one of the early clues to the crossover concept. And that was just gas exchange. And I think, in terms of students working in a lab, don't be afraid to go in there and get on a treadmill and get on the bike and test some idea out. You'll find something new that somebody overlooked. You'll find a new way to do something. Or even
I love that.
Yep. Even if you find what people have found in the 1930s, it'll be in your mind. You won't forget it.
Was he actually able to finish the marathon after not running for 2 years?
Yeah. But again, he was a 144 guy. And he told me in Australia, his running club, they would run 400 meters. And they would run 100 of them in one session. It would take all day. But they were all in 60 seconds. So we used to say if you could run 4 440s in 60 seconds with 60 seconds in between, you could break 4 minutes. And these guys were out there, on a dirt track. And probably it looked like the beach after a while. Imagine 20 guys out there pacing on a dirt track. So that's how tough he was.
That is by far not a traditional 800 workout. I'll tell you, 100 400s. I was an 800er back in college as well. And I think the max that we would do on 4s or 440s was like 6 to 8 of them maybe in one training session. So I don't know how they maxed at 100 400s at 60 seconds a piece.
That's 25 miles of quarters.
Wow. He must have been an extraordinary runner.
Well, he was 144.
And the best.
So they did not take, I did some of those workouts myself. So you might take 60 seconds before the next one, and then maybe it's 2 minutes, and then maybe you jog a half a lap. So maybe it's 3 minutes. But they could maintain that.
When's the last time you did that?
Hmm?
When's the last time you did exercise at intensity?
Oh, I broke up.
Oh, might be man. When's the last time you did an exercise of that intensity?
About 50 years ago.
What? I have some sun. I can't see, but you look 45 beyond there. But
Well,
anyway,
I did train on my ergometer this morning for an hour.
there we go. What did I train for now?
The Milan, the race from Paris to Milan is on, to Nice.
Yeah.
Paris-Nice is on. And I won it this morning.
Oh man, that's beyond me. Yeah, that's crazy.
No, I do the last hour with it, watching TV.
Good. I did as well. How was it at Berkeley versus the other places that you have been? Because I've been pretty interested in Berkeley just because I'm very upfront, I guess. But how is it at Berkeley, just in general, the culture?
Well, this is a great place.
Yeah.
Each place I've been is different. So at Michigan, I worked in physiology and also in zoology. And Michigan has a medical school, and Michigan is a great school. And their current kinesiology department has taken over the old natural science building. So, anybody want to go on for PhD, look at the PhD program at the University of Michigan.
Why I asked that, I first of all, I love your because my best friend, he's a biochem major, and him and I. So first of all, he's astronomically smarter than me. He always been, and that's where that our butting heads comes from. But he got accepted to he got accepted to the University of Michigan, and so that kind of prompted me towards that direction. And then my direction after that wanted to go to Berkeley. So it's funny that you your legs in both universities, but I did not know that they were as far as you say that they are, Michigan wise.
Yeah. So it's a former dean here at Berkeley, who's now the president of University of Michigan. And he's an MD, PhD, so he understands organically the value of physical activity. And so Michigan has made a huge investment in terms of kinesiology. But also I could talk about Cal, because I talked to Kristen about, maybe coming here because she's interested in biomechanics. So this is integrative biology department. It's not a kinesiology department. So the biomechanics here are of birds, are of cockroaches, of worms, just basic biomechanics. We did used to have human biomechanists on faculty, but they left to go elsewhere. And I think we're going to have a job opening this year in biomechanics. And it might be, well, it'll be any kind of biomechanics. But when people apply here, the expectation is that if they're admitted, they'll get graduate student support for 5 years. Now, it's not free, but you'll have to be a graduate student instructor for a minimum of a year. And then faculty have research grants. And so I have several students, and I'll support them as graduate student researchers, so they'll work on their own research. So when you apply to graduate schools, as Amanda said, you read the literature, you find out who's doing what you want to do, and you apply there. Sometimes a student will come to see me and say, well, I want to go to school in California. And that tells me right away they're not serious. If they want to go to study with somebody who's really doing what they are interested in and what they project to be the future, they have to go there. Isn't that right, Matt? You have to be ready to go.
I had to find my mute button there, but yeah, no, you totally hit the nail on the head. If you're serious about it, you need to be willing to go wherever to work with who you think is going to be the most interesting or the best fit for you. I mean, I didn't do all the research I could have when I was looking at PhD programs, but it worked out fairly well for me, all things considered. But I can't stress that enough. If there's an area of research you are interested in, find out who's doing the work, find out what kind of work it is. Don't be afraid to reach out to people. You may have to send a couple of emails to get a response, but most people love to talk about their work, love to talk about their research, and but
have any of you folks, you're absolutely right, Matt. Has anybody thought about doing a PhD in Copenhagen, Denmark?
Funny you mention it. That's where I'm from, actually.
If I could go back.
Erasmus?
Yeah, I'm an international student from Denmark. I live near Copenhagen.
Yeah.
They're really good at exercise physiology.
Yeah, they're, I would say, amongst the world's best.
Yeah.
Or maybe they are the best. Because there are so many labs in the Panem Institute, in the Bispersberg Hospital, all around the city. It's just really an amazing place.
Yeah, and the August Crow Institute. That's where my dad studied for a while.
August Crow. Eric Richter is now the head of August Crow. Physiology and metabolism.
Okay, I didn't know that, but yeah.
Eric studied under Benck, didn't he? I don't think I said his name right.
Benck Saltine?
Yeah, Saltine.
No, I don't think not so much.
Okay.
I do think that Eric for a while studied with Hans Krebs.
So Dr. Brooks, you had kind of hinted at the crossover concept. That's not something we've talked about a ton in this class, although I think most of the students are somewhat familiar with it from their undergrad. But I was wondering if you could maybe just kind of explain the theory behind the crossover concept, if you wouldn't mind.
Yeah, so one of the, at the time, there was a lot of interest in what you would call energy substrate partitioning, how the body decides how much fat, how much carbohydrate, where and when. And again, from the first experiment on running the treadmill, I knew that carbohydrate was the basis of most muscle exercises. And then you could trace that all the way back to the 1930s. People made those measurements. They could tell that the combustion ratio was close to 1 for most activities. So that's mostly carbohydrate combustion. But as you know, we developed our interest in lactate metabolism. We developed isotope tracer technology. First studies on rats with tritiated, it's radioactive hydrogen isotope, and C14 radioactive carbon isotope. Not really appropriate for human experiments, but we did plenty of studies on rats, and that's how we really basically discovered the lactate shuttle. And then we could see that, you know, what training really did was it really didn't decrease the production of lactate at all, but really allowed you actually to, training allowed you to work harder, so actually you produced more lactate, but you burned it. So hard exercise is carbohydrate burning, and a large part of that is carbohydrate that filters through lactate and then gets distributed and oxidized that way, either directly or indirectly after going to glucose and then getting oxidized. So then when stable isotopes came along, we could do studies on humans, and that was really revealing. And you could just see how energy substrate partitioning would shift the balance of how exercise would shift the balance of energy substrate partitioning from basically lipid to lactate. Matt, if you make me a screen, I'll bring up a draft of a paper I'm working on.
Yes, let's do it.
Sure, go for it. You should be, yep, should be good. Yeah, you actually just had one published, another review paper published in the Journal of Physiology I saw.
Yeah.
In addition to the AT one. So I'll send that out to you guys later today.
Okay. Looking in my folders. It's always amazing me, somebody on TV, they hack somebody's computer, they go right into where they find exactly what they want to do.
Right.
And try to do that on your
No, my folders are organized. I just never remember where I save stuff.
yeah. Like, here's a paper that's been favorably reviewed, is not accepted yet. It's going to be in Frontiers. Let's see. Okay, so where are we? Am I going to be able to share the screen with you guys?
Should be able to.
Okay. I think this is it.
Yep, there we go.
Here we go. So this is a figure. Oh, so one of the things we talked about was passion for what you want to do. So this is a picture from a cardiac catheterization lab at UC San Francisco. And one of the doctors there got interested in lactate metabolism in the heart. So we took our bicycle ergometer and we put it over the cardiac catheterization table. And then the physicians there were able to put in a number of venous lines at this point over here and put one catheter in sort of in the middle of the biceps to represent an inactive muscle because the legs were going to be exercising. And one tube or catheter went up the arm, through the chambers of the heart, down into the hepatic vein, and one went all the way up and down the body into the legs and the iliac vein, and one went into the coronary sinus, which represents the venous drainage of the heart muscle into the right atrium. And over here are arterial samples. So we have 1, 2, 3, 4 catheters in this one vein over here, going to different parts of the body, and one in an artery over here. And exercising and study at rest. And so this is a result. This was me on that table. And here's my lactate level at rest, less than 1 millimolar. And fatty acids less than 1 millimolar. And here's the fuel used by the heart. 80% of the fuel for the heart at rest is fatty acids, and 4% fat, 13% lactate. But lactate's pretty low. So when you start exercising, you're at 40% of max. The lactate now is most of the fuel for the heart. And this is spill of lactate from the legs and other tissues into the circulation. The heart's sucking it up. And 4% glucose, and now fatty acids are down to 40%. So why do I show you this? This is an example of crossover. It's also an example of the lactate shuttle. The lactate becomes a major fuel, and so instead of this heart muscle being fatty acid dependent, is now lactate and glucose or carbohydrate dependent. So that's crossover. It's also the lactate shuttle. And then this is an example of you got to be willing to do it.
And you said that on the table right there.
Yeah.
That's awesome. That's so awesome.
We had a question if it's difficult for the to feed the catheters in, like if it's difficult to get lost in the circulation when you're inserting a catheter.
What did they used to say in the ancient world? All roads lead to where?
I'll let
To Rome.
Answer that one. Yep.
So if you find a large vein in your arm, you put the tube, it'll just go up into the heart.
They use X-rays to check the positioning, don't they?
Yeah, right. And actually, you'll have pressure readings the whole way, so you'll know when you start to see the atrial pulse. You'll know where you are. It's a bit of a U-turn almost to get into the coronary sinus. But the coronary sinus is right up against the VA node. So when you try to stick the catheter into the coronary sinus, you hit the VA node, and makes for some really interesting EKGs. Yeah, so then up the arm, down, keep pushing the catheter in. You can see it on their fluoroscopy, which they had, but they basically knew by pressures. And then they could tell when they were in the right atrium and then just it's a straight shot down to the inferior vena cava. And then under fluoroscopy, you can thread it into the hepatic vein, and then otherwise you can just keep pushing and it goes down into the leg. So as we used to say, if you don't go home with a few Band-Aids, you weren't in a physiology study.
There you go. I still
I
got
love that.
Lars.
Yeah, I love totally love that. That's like honestly how I've lived life, and especially where I come from. That's like how everybody uses things in general, right? Like if you don't put in the work, then like why should I trust you? Is where I come from. I don't know.
Yeah, well, it used to be an old adage we had: if you're willing to want to do this experiment and you get human use approval, you go first.
Yes, yes, yes. That's like love
Because
that.
the thinking was that if you're not willing to do it, you wouldn't do it to somebody else or somebody else's child.
Right, right. Yeah, I love it. Yeah, absolutely.
Now you have to get special permission to test yourself around here. You have to prove you're not biased.
Yeah, they call it a conflict of interest.
Yeah, it's as if, Matt, you could change the data by just wishing.
If only, right? Yeah, we did a little case study a couple of years ago, myself and two of the grad students I was working with at the time, and because the three of us were the subjects, yeah, it got punted fairly quickly from a couple of journals. So how can you be sure there was no conflict of interest or bias? I'm like, the data's the data, man. But so you
Well, it's
mentioned
also why I'm here though too. So like, so I come from I come from a gang environment, and it's like it's really interesting because from that type of a environment, you have to your worth. You have like, how do you substantiate your argument? So I come into the academic aspect of it, and it seems to be the same exact thing. However, there's also that conflict of interest thing, but it's like if my sole purpose was come in to just prove that aspect of it, is my being here within itself just a conflict of interest, or is that me trying to provide data for the greater whole?
Yeah. So a bigger discussion is what is bias? How does bias affect our lives in general? And so scientists, I think, traditionally have been amongst the best people to take an objective stance on issues, like the null hypothesis. There is no difference. And then you have to disprove the null hypothesis and obtain data to support some other alternative hypothesis. So bias, yeah. Well, here, hard data, it's hard to have bias. But so the feeling now is that is because in the past people were biased and did things they shouldn't have done, that bias needs to be minimized, the possibility of bias. And I don't disagree with that. I'm just telling you, when I was going through the ranks, if you were going to do it to somebody else, you had to do it to yourself. Okay, Matt, you had another question or point to make?
Yeah, I was just going to follow up on the stable isotopes. So I'm a little bit familiar, but I don't think any of the students are really quite familiar with how those exactly work. So maybe if we could just talk about how you've incorporated the stable isotope work into your research to find what you've found.
Yeah. So this is actually in chapter 10 in the textbook. And the 5th edition is out. It has a green cover. It's it's available on Amazon. So when you measure, let's use the analogy of VO2 max. VO2 is the rate of oxygen consumption. It's a rate. It's not the amount of oxygen in your expired breath. It's not a percentage, it's a rate. So the blood lactate level, the blood glucose level is the rate, is a resultant of the rates of production and removal. So we insist, we maintain that you can't know the rate of production of anything unless you label it. So you look at blood sugar, it's basically 5 millimolar. In the morning, varies a little bit after you eat, goes back down to 5 millimolar. When you exercise, in the Tour de France, it's 5 millimolar. So you could look at that and you say, well, nothing happened. That's absurd. There's a continuous production and removal. And so the same thing with lactate. Lactate is high. Why is it high? Well, because I couldn't remove it. Or maybe I produced too much. You can't know. So I do look at most of the studies in the literature that report lactate levels, and I chuckle. Was it overproduction or lack of removal? And incidentally, we have a paper that came out previously in the Journal of Physiology. First author is David Poole. And comment on it was by Rinaldo Bellamo, a noted physician scientist, who basically said that the treatment of sepsis is archaic and arcane. Sepsis is a very, very severe disease, which there's a lactate accumulation. But he's been challenging clinicians to show that sepsis is due to overproduction. So he said, basically, his conclusion of his article and his commentary on our article was that clinicians need to work with exercise physiologists, because we know that the level of lactate in any condition, including sepsis, is the balance of production and removal. And just because the lactate's high, that doesn't mean that there was an overproduction. It's likely a lack of removal. So isotopes are any kind of isotope is usually of in the environment in low abundance, but carbon-13 is what we use to study these days. Carbon-13 is heavy by 1 mass unit over carbon-12. So anything that comes from nature, like I don't have a piece of wood here, but I have some paper, which is from wood. About 1% of this is heavy carbon-13. But these atoms can be extracted and it can be put into glucose. So we can isotopically label glucose. Now, as glucose goes through the blood, we can track it, we can trace it. So that's why they're called tracers. And so if the product of glucose that I eat that has carbon-13 appears in my breath, I know I burned it. If I put 13C labeled lactate into the blood and it comes out in the breath, I know it was burned. And also if the lactate is incorporated into glucose in the Cori cycle, then the glucose that's the result will be labeled. So we can trace the course of metabolism. And as in this study here, we can put a tracer in on the venous side, and then or the arterial side, or one or the other. And then when the venous blood comes out, we can know what went in and what came out. So in these studies on the heart, basically all the lactate that goes into the heart, all the labeled lactate that goes into the heart comes out as CO2. So that means the percent burning of lactate in the heart is 100%. And the same thing we can see in muscle. Working muscle for a trained person, all the lactate that goes in comes out as CO2. It's all burned. So putting a label on something allows you to see the movement. Another analogy, let's say there's a town of San Marcos, right? I guess there's a town of San Marcos. And let's say there are 50,000 people in San Marcos who live there. So we take a census in January, take one in December, And there's still 50,000 people. So you could say, well, nothing happened in San Marcos. But 10,000 people moved in, 20,000 people were buried, 5,000 people died, and 15,000 people left town to go to another city. So just by looking at the level, you don't really understand what's going on. You need to put a tracer in there. So if we have an isotopic labeled person that goes into that mix, then we can follow him or her. And of course, when we do tracer studies, one of the rules is we can't change the concentration, so we can't put it in so much that we raise the level, but we can tag, and we put in actually millions of atoms that go into a tracer dose, and we can find out what goes in, comes out as CO2, and what goes in the form of glucose made from lactate gets oxidized. We can know the rate of gluconeogenesis, and when we put catheters in, as in this study, we can actually not just say it happened in the body, but it happened in the muscle, it happened in the heart. We did it in the brain too at UCLA. So by labeling these atoms, you can trace where they start and where they end up.
Yeah, and I don't know if anybody watched or not watched, if anybody looked at the slides that I put up from the little fat physiology talk that we didn't get time to have, but that'll kind of come back in the next couple weeks when we talk a little bit more about fuel selection during exercise. But this same idea of using tracers has not only been used by Dr. Brooks with glucose and lactate. A group in the UK led by Keith Frayne has also used tracers to study fat cell turnover. So basically, how do fat cells respond to an increased caloric intake? How do they respond to weight loss? What changes inside an adipocyte? And so the tracer concept, or I guess technique's the better term for it. It really allows you to take a look at what's going on inside the body without, having to do something invasive like a biopsy.
So some of the tracers we're working with now, Matt, are deuterium oxide, D2O, heavy water. And a colleague, Mark Hellerstein on campus here, is a leader in the world on this. So if you label water, there are always exchange reactions in almost every process. And so if you have somebody drink like a cup of heavy water, that water will mix with their body water, and anytime there's a reaction. So we're using to measure gluconeogenesis. So in going from the precursors to glucose into glucose, if there's gluconeogenesis going on, some of the reactions are in equilibrium with body water, which then has deuterium in it. So basically, the greater the rate of gluconeogenesis, the greater the rate of deuterium or heavy hydrogen into glucose. But also he's using it to study particular proteins, and the proteins in particular receptors, different body compartments. It's really incredible to use this tracer. Now, it is heavy water, deuterium. It's not radioactive. So it can be infused. It can be, you can drink it, and then you can sample body fluids. You can take urine, blood, tears. You can really find out what's going on. And so Matt's point about you can measure protein turnover or fat turnover is true. In the future, with mass spectrometry, we'll be able to measure the turnover of particular enzymes.
That would be next level.
Yeah. Well, we always want to go to the next level, don't we?
That's true. We do want to evolve. But one of the other things you could do with a slightly different version of the Viridium, or maybe, well, it's called doubly labeled water. I forget the oxygen isotope, but
yeah, O18.
Yeah, you can use doubly labeled water. You can give participants a bolus of it, and then over a 7 to 14 day period, have them collect their urine. And as your body water stores turn over, so you get rid of old water, you bring in new water, the isotopes will show up in the urine, and then you can use that to estimate carbon dioxide production, which you can then back calculate to estimate somebody's energy expenditure. Just as like a, you know, a not real world, but a clinical kind of example of when an isotope or a tracer study or anything where you're doing kind of a turnover. And so that was something, it was before my time as a postdoc in Kansas, but for a number of weight loss studies that they conducted there in the early 2000s, they were pretty much using doubly labeled water for every study to look at the baseline for their participants. How much energy were they burning before the intervention, and then how much energy were they burning after the intervention? So the application for these tools and techniques is obviously quite broad.
Yeah. So that's actually, you could get the metabolic rate. The old way was factorial, where somebody would say, okay, I played basketball 30 minutes and I walked for 2 minutes and then I ate lunch. You write all these things down, and then you go to tables and you get the VO2 for sitting and walking and playing basketball. And, you know, if you play like Kobe Bryant, it's a different level of metabolism than if we play. But anyhow, you can get an idea from the so-called factorial method.
Anybody know what those tables are called? It might be something a lot of you probably have not heard of, but it's known as the Compendium of Physical Activities. It was made by, I believe, Barbara Ainsworth from, she might have finally retired, but she was at Arizona State, I think.
Yeah, Barbara's alive and well.
I knew she was alive. I wasn't sure if she had retired or if she was still at ASU.
She's still affiliated with ASU, but last time I corresponded with her, was working pre-COVID a lot in China.
Oh.
I think she's great over there.
Yeah.
And this compendium of physical activity, of course, yeah, she was one of the prime movers in that.
Yeah, and so what this compendium is, guys, is that they pretty much surveyed every paper that had published the oxygen cost of given activities. And by given activities, I mean pretty much anything and everything from desk work to, you know, high-speed running or high-speed cycling or what have you. And so this database lets you go, and if somebody was to give you a handwritten log of their physical activities, which some people still do, before accelerometers and fitness trackers and step counters and all that, this is how you estimated what somebody did during the day, is you'd have them write down what they did, and then you could go to this compendium, find the activity, and if they told you what they did and how long, then you could estimate what the oxygen and energy cost were. As, come on, Roscoe, get out of the way.
Yeah,
it's in the way.
Let's use a lot of dietetics to write to create diets for people doing different activities.
Yep.
Amanda, I haven't heard from you. How are you doing?
Pretty good. I also had, I guess I was waiting for a pausing time, because my question for you was kind of a little bit different. I was curious, for someone who has been teaching for so long, like traditionally, how it has been for you to transition now with the pandemic. You know, if you're teaching on Zoom, what's it like for you, or what are the challenges, or how you're transitioning?
Yeah, so right now I'm teaching human physiology with a colleague whose name is Jose Vasquez Medina, my young, outstanding colleague. You know, the way I describe Jose is, do you know what the Queen of England's crown looks like? You ever seen that?
No.
Well, in the top, right in the front, there's a big jewel. That's Jose.
Ooh,
Yeah,
what a compliment. Hey.
So we're teaching physiology, and we teach it over Zoom, and we try to convey enthusiasm and passion for the subject. But we really would rather teach and be amongst the class and walk around and get people involved. That's a whole art.
Is
So
conveying.
how I adopted probably as good as anybody else. But you know, interacting with students in the flesh has its pros and cons. So it's a true story. Right? You can make mistakes. So for my 60th birthday, my wife Rosemary wrote to a lot of my former students to write letters, you know. And so I remember this moment distinctly. I won't mention the person's last name, but his first name was Brian. And Brian was in my class, and Brian was getting C's. But his family situation was bad. And as an instructor, you can read people's eyes, and you can tell who's awake and who's not only following every word because they're anticipating your words. So it was a histological section, fast and slow muscle, which was a big deal in those days. So I asked Brian to come up and diagram histological section through fast and slow muscle. And I only did that because I was certain he could do it. So in his letter, he wrote that for years he hated my guts for doing that to him, embarrassing it, putting him under the spot. And now he's a professor. He's an MD, PhD at one of the Harvard medical schools. He said, I do the same thing. If I have a good fellow, research fellow, I ask them to lead the discussion. And at first they're angry with me, and after that, they appreciate that I am having confidence in them. Yeah, so I really miss those, face-to-face interactions. But right now, we have to defeat this virus. We have to behave ourselves. And I'm in my office, so I don't need this. But the rest of the day, even though I've been vaccinated, I go around socially distanced, and it's kind of weird. We're in the Valley Life Science building, and it's like there's a plague, and then nobody's here. They're all home. But if you like people, you don't even have to like people as much as you like the subject, and you can convey to people your enthusiasm for the subject. I mean, so when I was getting my PhD, I had to take a course in education, and there was one course that we couldn't understand. The instructor who's basically saying you have to find out where the student is. You spend most of your time finding out where the student is. It's like going from A to B. First, you have to find out where A is. So you have to interact with the students to find out where they are. And then depending on where they are in their place in life and their situation, before you can lead them forward, you have to find out. You have to relate, and you have to understand. And then you can go forward. Otherwise, you're just wasting everybody's time. So do you teach down there at San Marcos, Amanda?
I wish maybe perhaps one day. I teach more like group fitness, and I teach like adults how to exercise. So one day hopefully I can teach kinesiology courses or something.
Yeah.
But as a student, I'm the kind of in class, I like to sit in the front row, and I like to make eye contact with the teacher the entire time. I think I'm easily distracted, so I like to remove my distractions by being up at the front. And I'm the kind of student that I constantly ask questions. I like to talk with the teacher frequently, as much as possible, if anything, a lot more than other students. And so it's been really challenging for me being on Zoom. I really prefer to be around people. I feel like I'm very much a people person. When I'm around people, I have more energy. And when I'm removed from people, I feel like I don't have as much energy. So I really feel like I cannot wait to get back into the classroom.
Yeah, I think you make a really good professor. I don't even know you, but I totally agree. You make a really good professor.
That's what I.
Oh, thank you so much.
Yeah, I just had a conversation with somebody like Amanda the other day. A student comes in, and they want to declare the IB major, and it's not kinesiology. I mean, there's physiology and there's exercise physiology, but there's 100 other courses, and it has to do with ecology and environment. So I get most of the pre-meds. So I was talking to this young man, and he was not doing so well in school, but I could tell he had passion. So I said, Marco, you should become a teacher. And I didn't want to say to him, you're not going to get into med school. You could be really valuable as a teacher. And who knows, you can from there, you can go anywhere. I use a baseball analogy. First you have to get to first. First you have to get to first. So my wife and I have a friend and colleague. He was a PE major, and he became an athletic trainer. And then he got drafted into the Army. And then they sent him to school to become a physical therapist. And he went back, and then he served in Iraq at the time. And he became a surgeon. So now he's 50 years old. He's finally gotten to where he wants to be, but he didn't know where he wanted to be. And but I could tell. I don't know Sebastian very well, but both he and I could tell you would be a marvelous teacher.
Thank you so much.
Yeah.
That's definitely my aspirations.
And you don't need
Well,
isotopes to become a great teacher, Amanda. You can do it without isotopes.
Yeah, they add up too. They're not exactly cheap.
Yeah, well, carbon-13 labeled lactate is $1,000 a gram, and one experiment will use about 25 grams.
I'm in the wrong industry.
Yeah, I think we used a medium chain fatty acid that had been labeled with carbon-13 for one of my PhD studies, and I think it was $500 for a gram.
Mm-hmm.
Just adds up quick.
Yeah, the thing about lactate is it turns over so fast, most of it comes right up by eating the breath. So you have to give a whopping dose basically to have something measurable in the blood.
Yeah.
Fatty acid turns over slower. So
The dose was pretty small. I don't remember, but
yeah.
Do you think within those, so this is going to be my question, just because it's loosely associated with just my research, is within your area of expertise, do you think, or have you seen any differences within race, ethnicity in the substrate consumption, substrate use, anything along those lines? between ethnicity, just differences in general.
No, we haven't, honestly, we haven't looked.
I appreciate your answer.
So the NIH requires us to do studies on men and women. And then it's expected that our study population will reflect the local population. Okay.
Yep, I find that like super interesting because I read another study that looked at the population just within California, like San Diego, California. And if you look within California, it's like the population within California is so diverse that it would be more advantageous for you to look at or at least take into account like the race, ethnicity, and the studies that I've seen, they don't even look at that. Like they don't or look at that, like that's not their question, right? That's not their research question. It's not even taken into account.
Well, you want your population to be representative. Your study population, your study sample to be representative of the population. But I think the best we can do right now is to acknowledge that our data are limited. So for instance, Dr. Schubert was talking about studies of fat metabolism and using doubly labeled water. So I was on the macronutrient panel that did the previous generation of studies to define the RDAs. And so we had access to every doubly labeled water study done at that time. And there were about 500 studies, but they were all done in Europe and in the United States. And they were all done in medical schools or basic science departments. So almost all the subjects were of European extraction. So we had to say, this is our results are interpreted to be based on this population, and are probably not representative of the whole U.S. population. But right now we're frankly in a stage where we need to be inclusive, and that including large groups of people and doing studies, and then maybe on a post hoc basis try to work out differences between men and women and people of different ethnicities. I'm not interested in finding differences right now. Similarities, yes.
Well, and I think it also kind of comes back to the bigger picture of you're studying physiological phenomena, physiological constructs, physiological processes, and mostly interested in how they operate and whatnot. And I think in some areas where we're still kind of figuring out these mechanisms, we're not at a point where we can really afford to bring in the ethnicity question yet, because we're still developing the data, still developing the knowledge.
We're developing the basic concepts, but it was mentioned, my wife is a clinician, and she understands that different ethnicities respond to certain drugs differently. And then as a clinician, you have to know that. So if you want to treat somebody for immunological response, if somebody is of African background, you might have to double the dose of cyclosporin. And not conclude that if you give somebody a standard dose, that they're not a non-responder. No, you haven't given enough medicine. And so that's probably the future of medicine. There's a lot of talk about individualized, personalized medicine, looking at the genomic code and saying, okay, regardless of your ethnicity, as an individual, you'll respond to this drug or you need this dose. And That's in the future. Precision, it's called precision medicine, where you dial in the dose depending on the person's genes.
Do you think we'll ever get there with exercise? Because I know there are researchers who like to think that that potentially is the future.
Yeah, people are selling that. But nobody, the science is not there.
Yeah, I personally don't buy it myself.
I don't.
So does it come back to then if, let's go back to like exercise for weight loss, for example. So if an exercise intervention doesn't have maybe the changes in weight that we would expect to see, do you think that's because it's a failure of the dose? Or can we not jump to that kind of, well, I guess I'm kind of stretching a bit. That would be a bit of a jump to a conclusion, but you know, you mentioned it with responders versus non-responders to medication, and then there's this very, it's not a small group, but you know, there's this group of researchers out there who are pushing that that non-responder responder theory in terms of exercise. And is that, where do you sit on that kind of, do you feel there are non-responders to exercise, or that they just should do more?
Everybody responds, but the degree to which they respond varies. So I was impressed when I turned in that you were talking about power output, and I got to see some data, privileged data, on the blood lactate response of Tadej Pogačar, who won the Tour de France. And at 6 watts per kilogram, you know what his blood lactate was? 1.
1.
It's basically resting. So anybody else would have been 4, 5, 6. Some guys 10. He's 1. So are there individual differences? Yeah, and they're usually beautiful, aren't they?
That's insane.
Well,
That's
then I guess,
crazy.
yeah, then the question becomes, how do you take those physiological freaks, and is there something that we can learn from them to apply to a clinical population?
Sure. And that's I talked about my colleague Vasquez Medina. He studies elephant seals. And if you ever seen one, they are super fat. They look like a blob. When they come out on the beach, they look like a turtle. They're that fat and they just sprout blubber. They don't have heart disease. And they can dive down in the ocean, go hundreds of meters down, and to where the partial pressure of oxygen causes free radical generation like crazy. And they have no damage. So you're right. We can, if we could find an extraordinary athlete and then learn from him or her, that's great. The comparative physiologists, that's what they do. Do you guys know the Krog principle? August Krog? Where's Rasmus? Rasmus, what's the Krog principle? August Krog.
I actually don't recall it. I've definitely heard of it, but I don't really know. Actually.
So basically, whatever experiment we could design, nature's already done it.
Oh.
So there's an animal model of every process we want to study.
I like that. It's pretty interesting.
It's called the Krog principle.
Interesting, yeah.
Yeah, so there's been so many evolutionary forces on animals through the course of history that animals adapt in different ways. And so there's a model, it's typically an animal model for any phenomenon you want to study.
So, like sleigh dogs.
What about sleigh dogs? They burn fat like crazy, don't they?
Insane. They have such great just cardiovascular just systems. Oh man, it's very impressive. Not to say that without like us. Never mind. I'm not gonna say anything. Yeah, that's cool.
So,
I have a question actually. Um,
go for
so of course I read the two papers that were assigned for today. One to review the tortuous path of lactate shuttle discovery, and then also one of your studies on traumatic brain injury.
Mhm.
And then I actually also read your review in Cell Metabolism, the Science and Translation of Lactate Shuttle Theory. And so I knew that, lactate is not just a byproduct like we used to think, and it's actually a really important energy source for muscles and other tissues. But I didn't know it had that many implications in things like looking at the abstract here, resuscitation from acidosis, treatment of traumatic brain injury, maintenance of glycemia, reduction in inflammation, cardiac support, heart failure, like heart attack, cognition, and even cancer and sepsis. Sepsis I know because I had that a couple of years ago. And
You had sepsis?
yeah, and my lactate was high. And I remember my mom was so concerned with it because she is a chemical engineer, and she actually develops the sensors that they use, or the analyzers they use in the ICU to measure blood gases and lactate. You might have even used some of that in your research, analysis from radiometer.
Yeah, we use radiometer stuff.
Yeah, that's where she works in
That's the
in
class.
Copenhagen.
That's the class of the field.
Yeah, so yeah, anyways, I digress. So I was wondering, the topic of lactate, if someone wanted to do a PhD, would lactate be like a hot topic or a good place to study? Is there a lot of a good outlook
The people who
in
do
that
that
field?
Copenhagen,
It more so in like the biomedical science direction, more so than exercise?
yeah.
Okay.
So again, look in the literature and you'll find there's a lot of activity in Copenhagen.
Yes, that was my second question is, do you know where in the world that is very popular at the moment?
Yeah, so there aren't a lot of people who actually can do the isotope tracer technology. There's some in
Interesting.
Berkeley, and there's some in Copenhagen. There's some in Australia.
Okay, interesting. Thank you.
Yeah. So biomedical adaptations of tracer technology. I think there's a dearth of people who can do that, and it would be great to learn those skills.
always, that's another thing to kind of consider for you students is if you do decide to go on to do a PhD, look at the, not only read literature, but look at the research techniques that your potential mentors are using. Are there things that you want to learn or get hands-on experience with, for example? So like, I got phlebotomy certified during my PhD, so I could take venous blood. Instead of just doing capillary sticks and getting little drops, I wanted to actually get, containers so that I could do multiple analyses. But if you're interested in muscle physiology and you want to learn to do muscle biopsies, find somebody who's doing that. I mean, there aren't as many of them as there were 20, 30 years ago, but people are still doing biopsy stuff. If you're interested in physical activity, go and find somebody who does a lot of work with accelerometers or population data sets and things like that. So again, read the literature, look at what they're doing, and then you can tailor your experience to kind of fit what your goals or your objectives are.
Okay. So can I just ask one final question? Who else is going to present to your students, Matt?
Let's see. I've had so the next guest I have is Ed Mellinson, Colorado. You probably know Ed.
I know him. Yeah.
So yeah, he's going to talk about substrate, and then
Dr. Sabapathy already presented for Australia.
yeah, we had Dr. Abby Smith Ryan present the first week of class. She talked about body composition. She's at North Carolina. And then when we talked about VO2, we had Surendaran Sabapathy. He was one of my PhD mentors from Australia. Talk about, he was mostly going to talk about VO2 and critical power, but it kind of ended up being an aside into thermal physiology. But that's really the whole point of these interactions, is to give the students a chance to see and hear what other people are doing. So we'll have Ed, and then we'll have a colleague of mine talk about some of his hydration, dehydration work. His name's Chris Irwin. And then I'm kind of looking for like a neuro mechanics person to wrap up the semester. I haven't found a ideal person yet, but if you got anybody in mind.
Reggie Edgerton.
Edgerton?
Reggie Edgerton, yeah,
Okay.
at UCLA. And actually,
yeah.
and so, back to Amanda's point, this Zoom time really allows us to do what we're doing today. We don't have to fly around and rent a car, or all that kind of stuff. We just spend an hour together.
Yeah.
So, V.R. Edgerton. I don't know what the V stands for. He never told anybody, but Reggie. Okay.
Yeah. I don't mean to keep you longer than necessary. If you need to, we have a bit of extra time, but Dr. Brooks, if you need to head on out, then we can let you go.
And if anybody has a question they want to ask, now's the time.
Yeah, that'd be a good time for. So we did have a question in the chat asking, what are some new challenges to research or teaching that occur today but were not prevalent when you started?
Well, beyond Zoom, huh? So I think, when you get your master's or you get your PhD, you're probably going to be, for a period of time, the world's expert in that, which is good, but the world is going to change right under your feet. So you have to be forward thinking. You have to be aware of what's going on, about what the tools and techniques are used. So with Rasmus, we talked about tracers, and we also talked about tracer technology. The world is going into genomics, proteomics, metabolomics. And these are all techniques. CRISPR, another technique. But in 5 years, it'll all be passé. So the question is, what is the question? So some of this we touched on today. How do we use this information for the betterment of humankind? And over time, different tools will emerge, and then they'll be tested, and then the world will go on. So CRISPR is a great technique for gene modification, but as my students ask me all the time, well, can I cure this disease with CRISPR? In an adult? And the answer is no, you can't do that. You have to modify the organism at the embryo stage. And as far as I know, that's completely illegal and unethical. So what can we do? So CRISPR is all the rage, and Nobel Prize here at Berkeley. Everybody's very happy. Jennifer Doudna is articulate and beautiful, smart. Couldn't have a better poster person than her. But I think we all realize its time is now, and in 5 years it'll be something else. So if somebody's really technique oriented and dedicates themselves to a technique, you'll have a good run, but it won't last forever. You just have to keep moving. So we talked also about spectrometry, and what we're doing, but the world is moving ahead with deuterium labeling, tandem mass spectrometry, to not only measure metabolites, but look at the turnover of receptors, the insulin receptor. My gosh. Turn over the insulin receptor. How profound is that? And of course, that's related to exercise, and probably nobody's thought about doing that, looking at their insulin receptor turnover as a result of exercise in men and women, young women, people of different ethnicities. It's going to be possible.
Do you think we're close to having a wearable lactate sensor?
They exist.
Yeah, I knew the group down at, or I know the group, there's a group at UCSD that have been trying to build one for a while to measure lactate, glucose, and sodium from sweat, I think. But I don't know if anybody's quite at prime time yet. I don't think there's a commercial marketable yet.
There's one that can put in tattoos, I think, that can measure
I think I
something.
know what you're talking about.
It's like they put the, I don't know, sensors in tattoos and the ink, and then it can measure, I don't know if exactly lactate, but I think glucose is one of the things that they're considering or doing. I forget his name. He's at UCSD.
There's worldwide interest in wearable devices, in miniature devices, and you can find a paper in literature that was in Nature, big time journal. First author is Wei Gao, and we measured electrolytes and metabolites in sweat. So we measured glucose, lactate, some other metabolites. We measured sodium, potassium. These are all wearable devices. The problem is we've known for a long time that sweat doesn't always reflect what's in the blood.
Yep.
So some years ago, Google built a contact lens that could measure glucose, and therefore that was going to be the rage for treating diabetics. But physiologists knew for decades that tears glucose is not the same as plasma glucose. So where is that? Well, they dropped it because they found out it worked technically, but it didn't work for people. So the question is how to develop a sensor. But this is this field is burgeoning, and there are startups galore. And there is a company in San Diego that I consult for that makes these devices. And they're not all not on the market yet, but they're going to be devices. They're going to be wearable sensors that'll be FDA approved for diabetics, for use in the hospital as point of care devices, so you can actually watch the progress of a disease. I'm glad this came up. It's a really fantastic idea. But the key thing is wherever you put your probe, it has to track what's happening in the body, not just locally, like sweat. You can measure glucose and lactate in sweat, but that doesn't tell you. You couldn't manage a diabetic that way. Could not. But people are working on this. And some of you probably might go into biotech or high tech. And you might wind up actually doing this. So in diabetes, a big problem is diabetic ketoacidosis. So you would need a device that actually measured at least three things: glucose, lactate, and ketone bodies. And some of these devices are emerging. They're Bluetooth enabled.
Mm-hmm.
Talk to your phone. It was software that have been written to be able to anticipate bad events, or to warn patients about whether they need sugar or insulin, or or Cytomax, whatever.
Ah, you threw in the Cytomax plug. So how many of you students out there have heard of Cytomax? Anyone heard of Cytomax? So Cytomax is it's a sports nutrition company, but they first kind of got well known as a sports drink that included lactate in it as a carbohydrate source.
Yep. And you know, it was Jack Azevedo actually did a pilot study,
Jack
not a
did
pilot study, clinical study.
Yeah, comparing Cytomax to Gatorade, looking at the lactate production and clearance and the glucose rates of production. And it's been quite a few years since I've looked at it, but if I remember correctly, I think he saw a performance improvement with the Cytomax compared to Gatorade.
Yeah, he did. Because, so Cytomax was made by a company called Cytosport. Cytomax gave rise to this company, Cytosport. And they were very much interested in it until they started to manufacture muscle milk. But it was a family business, and they're just all great folks. But I knew that there were intestinal lactate transporters. And we also knew that there were cell membrane lactate transporters and mitochondrial lactate transporters. So rather than trying to supplement athletes with glucose and putting so much glucose in a drink that people got nauseous, we could utilize a broad profile of metabolites in a drink that would not compete with each other or become saturated in the amounts we put in. So so we designed Cytomax to contain a lactate polymer, which I invented, and some glucose, some fructose, and it was what Mr. Armstrong used. Of course, Mr. Armstrong used a lot of other stuff.
Yeah.
And anyhow, the company didn't really invest in advertising, so most of the students don't know about that product. But the company was very successful and sold to Hormel. Hormel is a big industrial food company that makes, you all had Hormel products. So you had Skippy peanut butter, you had canned hams, processed meats, salami, bologna, sausages, all that kind of stuff. They didn't really know what to do with Cytomax. I don't know if it's still made. My contract was up, and the company I worked with sold to another outfit, and I didn't really like it. So I'm independent now of that. But that's an example of what I say to my students and to my colleagues. Grant support here is really important, but if you can't walk out into your lab and think of something that people can use, you really aren't thinking very well. So I made Cytomax in my kitchen. Made a lactate polymer, just based on chemistry. It was easy.
I think that's the first time I've heard food chemistry called easy, but.
Well, I learned in chemistry that maybe you guys in OC have learned this, that the salt of an acid is a base. So if somebody has acidosis, you can give them sodium lactate. That will raise the pH right away.
Yeah, and isn't that a clinical or medical tool in certain situations? Is they'll give you a lactate IV?
Yep. For acidosis.
Yep.
And it's the L isomer of lactate. An early formulation was called lactated ringers, and it was a racemic mixture. So the enantiomer of L-lactate is D-lactate, and D-lactate is toxic. So the lactated ringers is used, and it's like one step forward, 2 steps forward, 1 step back, because it contains the D enantiomer. But as Dr. Schubert mentioned, that L-lactate infusion can be used to treat metabolic acidosis.
All right.
Hey guys, it's a great conversation. Good luck with your other speakers, and I think if you contact Dr. Edgerton, read about his most fantastic work on locomotion.
Yeah, I will definitely look into that because I'm trying to give him as broad an exposure as possible. So I appreciate the hat tip.
Yeah, and there are a lot of other people you can get as well. And also, I think this year, for the first time in a couple of years, I'll be attending the Southwest Regional ACSM Conference.
Where's that happening this year? I know it's not till October, but
it's in, where is it going to be? It's in your neck of the woods, guys. Let's see.
Cal State Long Beach.
No, it's north of there. Let me see if I can find my schedule.
I'm checking the website right now. It's last year's program. And just see a date. I don't know if they have a location yet. Oh, here it is.
Hilton, Orange County, Costa Mesa.
Costa Mesa.
Costa Mesa, yeah, that's it. It's a 1.5-day meeting, and students, you'll get a kick out of it.
Yeah, hopefully more than a few of you will be presenting a poster or something there in the fall. But it's a really good meeting, and it's relatively intimate as far as the ACSM type meetings go, because the national meeting can be a bit overwhelming in terms of the numbers, but the regional one, especially our chapter, is fairly, I mean, it's a big chapter, but in terms of the number of people that kind of show up, it's a bit more intimate, and so you have the opportunity to go up to people like Dr. Brooks here and just chat them up while you're looking at posters or whatever. So
yeah,
hopefully we'll be able to get a few of these students up there and meet you in October then.
It'll be my pleasure.
All right, well, we appreciate your time very much, and I hope you and your family continue to stay safe and healthy, and we will catch you again very soon.
Yeah, the same to you, Matt, and you and your family, and your family, your professional family too. And all these young, wonderful minds that you have under your tutelage.
Yeah, I'll try not to corrupt them too much.
Yeah, well, I think this course is a great idea, and the advantage you get in this case of Zoom is really amazing.
Yeah. Okay.
Your
great.
time is much appreciated. I
Thank
really
you.
appreciate it. Yeah, loved it.
Thank
Loved it.
you.
Thank you.
Guys,
Thank
take
you.
care.
Thank you so much.
Take care.
Bye.
That was so cool. Man, he was cool. I love that dude. That dude was sick.
Well, good. I'm glad you guys enjoyed that.
I was trying to be polite, but I really